Hi, my name is Angelica Vázquez Ortega. I'm an associate professor of geochemistry at Bowling Green State University in Ohio. Today's topic, the title is Characterizing and Comparing the Molecular Composition of Extractable Human Material from Organic Farm Soil and Lake Dredged Sediments. Okay, let's start defining what are dredged sediments. So dear reference, everybody here, we are located at the northwest Ohio, very close to Lake Erie. So the Western Lake Erie Basin, including the federal navigational channels in the Maumee River and a portion of the lake, are being dredged yearly. This is in the Toledo Harbor area. These navigational channels, are dredged yearly in order to ensure that cargo and different boats can navigate the area and provide the different services and goods in the area. Typically, the dredged sediment that it's being dredged has been disposed in the open waters of Lake Erie, but this can have negative impacts on the water quality of that area. For instance, the sediments are there being dumped. The nutrients within them can be redistributed in the water column, as well as affecting the benthic community. The redistribution of the nutrients can exacerbate harmful algal blooms that has been happening in the Western Lake Erie Basin for the past decades. For this reason, the state of Ohio prohibited the open disposal of dredge on July of 2020. Now, different federal and regional agencies are looking for beneficial use of this material. The specific beneficial use that I'm going to be discussing in this presentation is to use the dredged sediments as farm soil amendment. And my research group has been conducting research in greenhouse settings as well as field demonstration settings. So I'm going to show you some results obtained when corn was grown in a greenhouse setting. The experiment, we use corn as the crop, and we designed the experiment in buckets or mesocosms where the soil was either 100% farm soil, 100% dredge, or a mixture between the two. So we have a mixture of 95, 5, 90, 10, and 80, 20 of farm soil versus dredged sediments. And the farm soil used in this experiment was an organic farm soil that has been managed organically for more than 20 years. We have collected data for characterizing the chemistry and the physical characteristic of the soil in terms of the soil health, and also some information regarding the health of the crops and the yield. The data that I'm presenting here is indicating that as the higher the yield percentage in the buckets, also the higher is the above biomass or the corn height in this specific instance. We also observed that the root system was more robust. A larger amount of fine roots was observed as dredged sediment amount was also increasing. Regarding the soil chemistry, we observed that the cation change capacity in the soils increased as well. And it was due of the dredged sediment having a high content of calcium. Similar results have been observed for other crops, in specific soybean and specialty crops, including lettuce, tomato, and carrots. We also are conducting demonstration farm experiments where we are adding the dredged sediment in tons per acre, and we are growing corn and soybean in the area. In addition to measuring soil health and the yield of the crops, we are also measuring surface and subsurface runoff with the intention to quantify the export of nutrients when dredged sediments are used as farm soil amendments. Okay, so the research question for this presentation is, does the increase in soil health and yield associated with amending a farm soil with dredged sediment is influenced by the soil organic matter quantity and quality? So many different parameters can be influencing what we are observing in our research. The specific question here is organic matter having some influence. And we're going to be measuring how much in parts per million and also some more qualitative description of the organic matter. In order to answer this, or yes, to answer this objective, we're going to be employing a method. It's an organic sequential extraction that it's named Humeomics, that was developed by Piccolo and his peers, Nevioso and Drossos. So it's a very complex sequential extraction using organic compounds, but to simplify this, the products are fractions that are categorized aqueous soluble versus organosoluble fractions. And here it's all of that sequence of steps that needs to be taken. But if we look at this diagram, we can think of the soil organic matter as a supramolecular structure, that it's held together not by condensed carbon-carbon bonds, but instead by other intermolecular forces, including hydrophobic interactions, cation bridging, and so on. So here we have that specific example, and then these illustrations are describing how the sequential extraction is removing the different components, okay? So again, we have unbound and we have ester bound and ether and ester bounds. And these ester and ether compounds are being further breakdown, but no carbon-carbon bonds are break down by this sequential extraction. So this is the sequential extraction in pictures that I took pictures of Samira Proba when she was conducting this experiment in the lab. And all of the fractions were characterized with different methods. So we use the Shimatsu to analyze the total organic carbon, a discrete analyzer to measure total phosphorus, total nitrogen. We measure the functional groups using an FTIR. And then we also measure different components of the organic matter by fluorescence spectroscopy. And fluorescence spectroscopy was coupled with Parafat to actually deconvolute the specific components. And we use OpenFluor database to compare the components that were identified in the fractions, organo-soluble fractions or aqueous fractions with those found in the literature review. So this data is for the FTIR data that Proba characterized. So here we have, again, the FTIR characterization for the organosoluble and the hydrosoluble. We are comparing our results with those obtained by Drossos 2017. However, the discrepancy here is that he used GC and LC-MS to identify the different functional groups. They also use FTIR, and some of the FTIR groups that we identify are also in agreement with those found by Drossos. So the upper portion is the FTIR data for the organosoluble fractions, and the lower one is the FTIR data for the aqueous fraction. In general, what we found is that the dredged sediment organosoluble fractions contain saturated hydrocarbon and aromatic moieties, which can contribute to the supramolecular structure hydrophobic characteristic. It's also important to mention that having a high hydrophobicity degree in this organic matter, it's also very important for the formation of soil aggregates. And this tends to sequester soil organic matter over a longer period of time, allowing that organic matter to decompose and provide carbon to microbial organisms. We also have fluorescence spectroscopy data. And this data, the way that I'm presenting, it's named excitation emission matrices, where the sample is a non-destructive technique, where the sample is excited by different wavelengths, and then the emission in that pair of excitation emission is recorded, and then we draw this type of contour maps. These images or this fluorescence intensity can be classified in different regions. For instance, more microbial derived compounds versus more terrestrial compound derived compounds. For instance, humic and fulvic that are more recalcitrant and aromatic in nature. From this type of data, we can also measure different indices, for example, humification index and fluorescence index. And humification index values higher than 10 indicate a high degree of aromaticity and terrestrial origin, versus fluorescence index higher than 1.5 indicate that the organic matter has been microbially derived. When we calculate, or when Proba calculated the humification index and the fluorescence index in this data set, she observed that the humification index was very high, indicating terrestrial origin, and the fluorescence index was also high, indicating microbially derived. So the dredged sediments have both origins, are terrestrially derived and also microbially derived. And we have to think about this of the journey that the sediments are taking. So they are from the farmland, which is organic matter that is terrestrially derived. However, these dredged sediments move into the rivers and at the bottom of Lake Erie, acquiring a microbial derived signature as well. So we couple this with Parafact, which is a statistical analysis that deconvolute the different components. And we can observe that for the organosoluble fractions, we have components that are both terrestrial and microbial, and for the hydrosoluble as well. So the Parafact, it's in agreement with the humification index as well as the fluorescence index. So we also measure the carbon, total organic carbon, total phosphorus, and total nitrogen associated with the different fractions. And we can observe that all of the organosoluble fractions are very rich in total organic carbon. The hydrosoluble, the aqueous 2 is also very rich in total organic carbon. When we look at the nitrogen, the organosoluble 2 and 3 fractions are enriched in nitrogen, and we can look at the scale here. Also, the hydrosoluble fractions are very enriched in nitrogen. When we look at the phosphorus, it seems that the aqueous 2 is the fraction that has the highest amount of total P. So it is very important to characterize the different fractions that are going to be, as the soil organic matter is decomposed, they are going to be breaking down from that supramolecular structure and serving as electron sources or carbon sources, or also being associated with the nitrogen and the phosphorus. They're going to be releasing these nutrients into the pore water, being bioavailable to microorganisms and also to plants. So soil nutrient-containing compounds are very important to also maintaining the soil organic matter hydrophobicity. So the different fractions are giving the supramolecular structure different signatures, more aromatic, hydrophobic, and they're going to be releasing these nutrients slowly versus other fractions that are more labile, that can be more bioavailable more easily and more quickly to the plants and microorganisms. So this slide, it's a little bit crowded, but I'm going to walk you through. It's a comparison between the dredged sediments that were characterized by Proba and the soil organic matter that was characterized by Katarina Kiefer, another master student in my group. So we are comparing the different aspects that were measured between both organic farm soil and the dredged sediments. For instance, if we look at the nutrient content, it seems that the dredged sediments has the highest amount of TOC, and also higher amount of TP and TN were found in the aqueous 2 in the dredged sediments compared to the farm soil. So if we add dredged sediments to a farm soil, we are adding a higher degree of carbon and also a higher degree of TP and TN in the specific aqueous 2. So when aqueous 2 is bioavailable to the microorganisms, it might be releasing these nutrients into the soil. When we look at the fluorescence indices, the dredged sediment has a higher humification index, indicating that it's more humic and aromatic in character when compared to the organic farm soil. Also, the dredged sediment, in comparison to the farm soil, has in the FTIR, when we look at the FTIR, has more overtones, meaning having more of that aromatic characteristic, and also have nitrogen functional groups in the organic matter, more indicators. When we look at the parafact, they had very similar components, both terrestrial and microbial. In general, or we can say that the dredged sediments are highly humified and more aromatic than the organic farm soil, and the dredged sediments were more enriched in total P and total nitrogen in the aqueous 2 fractions. So we can say that amending an organic farm soil with the dredged sediments could beneficially increase the aromaticity and hydrophobicity of the farm soil, so organic matter within the farm soil. So to end, I have here a few agricultural implications where it seems that the organosoluble fractions in the dredged sediment, again, had a higher total organic carbon content, which this can help in decreasing the bulk density of the farm soil, also increasing the soil aggregation, making the soil more healthy when we look at the physical aspect of the soil health. When we think about the organic compounds within the dredged sediments, and when these are degraded, all of the organic matter and associated nutrients are going to be bioavailable again to plants and to microorganisms. Obviously, the more recalcitrant will be released slowly over time. Both have terrestrial and microbial, and again, those compounds that are more microbially derived will be more labile and provide these nutrients more quickly to the plants and soil biota. However, the more terrestrial compounds that are more aromatic and recalcitrant will degrade over a longer period of time, and this will provide a carbon source and electron source over a long period of time. The aqueous 2 fraction had the highest total P and total nitrogen content, which the composition of this fraction will again serve as a constant source of nutrient in agricultural systems if their sediment is it's an amendment that is being considered. Future research include characterize all of these organo and aqueous soluble fractions using mass spectroscopy. That's something that we are planning to do. And also, so far, we only have presented the dredged sediment and the anorganic farm soil. We want to use the Humeomics approach to more conventional farm soils, which is a more typical management in Northwest Ohio. And also mix this with different best management practices, such as incorporating cover crop and or maybe no-till and so on. So I want to say thank you to Samira Proba and Katarina Kiefer, which were my master's students working on this type of data, and also to my collaborator, Marius Drossos at the University of Basilica in Italy, which he has helped me to make sure that the Humeomics method is accurate and we are obtaining good results from this one. So thank you so much. If you have any question, please email me, and uh, that's it.